I'd like to use this first video just to welcome everyone to the microbiology class where we're going to be studying small life that is out there. And to sort of put things in perspective for you, I want you to think about different ways of looking at microbiology. And the example I pulled out here is a sort of old disease known as anthrax that we aren't too familiar with these days unless you're working as a vet or something like that. But if we look at these different ways of looking at anthrax here, as a microbiologist, we can look at anthrax in terms of the cells that are actually going to cause this disease. And these cells have a very characteristic shape that we can see in this electron micrograph right here. And those cells can be analyzed and look at a variety of different characteristics that define that particular cell. We can also grow these cells up in the laboratory as colonies, and here you can see some colonies that are growing on what's known as blood agar, and you can see they have a very characteristic size and shape and color and the edges to this. So both of these things have certain characteristics that are associated with anthrax in terms of the organism that causes it, growing as either single cells here or as colonies. But we can also look at this in terms of the disease, anthrax itself. And when these bacteria cells are able to get in through typically a tear or a break in your skin there, the bacteria will cause a disease that results in this formation of this black kind of tissue that's almost like charcoal in its consistency, kind of crumbly, like the briquettes that you would put into a barbecue. And that's actually where we get this name anthrax. It comes from the Greek word for coal. So just to think about this and put it in perspective, that when we look at microbiology, yeah, we are studying small life that is out there in terms of the cells, but we can study it at these different levels to see a whole variety of different kinds of characteristics that are associated with this term anthrax over here, whether it's the cells, the colonies, or the disease. When you're going through the material, one thing I would like to point out as you're reading through is the textbook does have a variety of things listed at the beginning of each of the parts of the chapter known as the learning objectives. And if you look at this, these are really the kinds of things that you should be using when you're studying. If you're taking notes from these lectures or writing notes from the book, these are the kinds of things that you really want to focus on and make sure that you can answer these kind of questions. And these are basic memorization kind of questions when we look at this term describe over here. You're going to have to basically recall these different kinds of things here. And so we'll take a look at some of these different things and I'll try and put them into perspective for you. And this first one we can see they're talking about having you describe how our ancestors improved food with the use of these invisible microbes that are out there. And when we look at this idea of these invisible microbes, they've been around for thousands of years, but nobody really understood the presence of these things because you couldn't see them. And what we now know today is that when you're looking at this process of fermentation, you are literally looking at microbes that are out there that have settled down on your food or your drink, and those microbes have eaten it, and they have produced waste products. And those waste products are what actually turns out to be toxic to other microbes that would end up spoiling your food in a way that we couldn't eat it. And over here we can see an example of some yeast cells known as Saccharomyces cerevisiae, and these guys probably landed on some grains or some ground up grains that are out there, and some people that were out there took that stuff and cooked it, and that's where we get our modern day bread here. The bread is rising because these yeast cells that are growing inside of this ground up grain right here has a whole bunch of sugars available, and the yeast cells are basically farting CO2 gas into there, and that's what's causing your bread to rise when you bake it. Some other people that were out there probably took some of this stuff and started drinking it, and then they started staggering around because there was a bunch of ethanol in there and they were getting drunk off of that. And here you can see that Even thousands of years ago, people were still using beer bongs, although this one looks like it's a little bit reversed right here. But more than likely, they were drinking their beer through these long straws to try and prevent getting a whole bunch of the solid, chunky stuff that was available there. If we look nowadays, we can see that there's a whole bunch of different kinds of foods that are being made by fermentation, or this controlled rotting of your food that's available. And depending on the type of microbe that is included there, you can get a whole variety of different kinds of things, from the acetic acid that you're going to find in vinegar, to the wine that has your ethanol from the yeast, something like fermented meats that are out there with salami, and things like cheese and pickles that we're familiar with, but then some of these other weird things. And when we look out there, we're talking about hundreds of different kinds of food products are actually rotten food, but it's rotten in a way that we actually enjoy the taste, and it does not cause too much harm to us, although some people would argue about wine and its effects there. When we look at this idea of fermentation and think about these foods are all rotten, well, think about all the other foods that are out there that are getting thrown out, that are also rotting out there. Those aren't necessarily going to be good things that you want to smell or eat. And this brings in this idea of disease that's out there, and that is definitely part of the field of microbiology, as we want to study these kinds of diseases that are out there. And some of the earliest kinds of recorded thoughts about what causes these diseases, one sort of idea that people had is that you had bad air from things like rotting organic materials, the trash and dinner from last night, and that gave us this miasma, the bad air, and there was something about that bad air that was causing these diseases. And you can also look at and think about, well, maybe it's not coming from the air. Maybe it's some change inside of you that's going off kilter there. Other people, you can go way back and look at these diseases are a punishment of the gods, or maybe there's a witch out there that used the evil eye on you, and that's why you're getting sick there, and that's why your family is getting sick as this disease spreads throughout your household. But again, it goes back to this idea that we can see this, but we didn't know what was behind that rotting there, because they couldn't actually visualize this stuff. But people did know that there were certain practices that you could use to try and reduce the occurrence of these diseases. And if you go back to ancient Rome and you look there, they actually had this huge sewer that we can see right here that basically came through the city, and it had rivers on both sides. So the water was coming in, people were bathing and drinking the water, and then it was flowing through the sewer and coming out on the opposite side of the town. And what they found is that sewers were able to actually reduce the spread of disease. And this leads to the question of why. And there you go back to that idea of, well, what's causing this? Is this something off with the fluids in my body? Did the gods curse me? Or is there some sort of bad air that's available? Well, and when we start to think about this idea of these sewers carrying away the waste that's out there, this is really one of the things where we can start to look at the numbers that are out there. And if you look at some of this data from the World Health Organization, you can see that we're looking at the leading causes of death worldwide out there. There are some other causes that fit into this sort of general category. A huge amount of these are going to be caused by cardiovascular tissue or cardiovascular conditions that are out there, but we can also look at this thing that is directly associated with microbiology, this idea of infectious diseases, from things that you're breathing in that are causing something like pneumonia to AIDS that's out there, or death by diarrhea and tuberculosis. And when we look at all these things, you're up above 20% of deaths that are occurring annually are the direct result of these kinds of infectious diseases that are out there. So this is definitely something that we want to study and learn a little bit more about what's going on in terms of causing these diseases that are resulting in death, but also finding ways that we can try and reduce that and reduce the transmission of these infectious diseases. The other kind of interesting thing is when people look at these things that are out there, and if you look at this cause of death right here, you can actually find that it does actually vary with your income level. And we want to take a look at these purplish colored bars right here, because this is showing the death by these communicable diseases. So things that can be transmitted like bacteria or viruses that are out there. And if you notice here, in high income countries like the United States, communicable diseases are not really resulting in a lot of deaths. The thing we have to worry about are these kind of chronic diseases. Again, things like problems with your heart that's out there. But as you work your way down and go into lower and lower income countries, what you find is there is a massive increase in the amount of deaths that are occurring as a result of infections that are out there. And so really, this is a very interesting discrepancy between these two things. So we want to identify what's causing this for ourselves, but we also want to identify what's causing this because microorganisms don't know about boundaries. They don't care if you're from a high or a low income country. They're going to cross that border and cause disease and death wherever they can. So it's definitely something that we want to learn a little bit more about these properties and what's going on with the microorganisms that are causing these infectious diseases. And when we go back and look at this idea, well, what's really needed in order to look at and study these infectious diseases that are out there? Well, remember, if it's miasma, it's bad air, but we can't see what's in the air. And that's where the invention of the microscope really played a key role in microbiology and defines it as a science, really, so that now we can start to look at the organisms that are out there. And if we look at go back to the late 1600s, there was actually a Dutch cloth merchant who wanted to look at the threads and fabrics, and he was able to grind his own magnifying glasses, and he made these little primitive kind of microscopes that are out there. You basically put your specimen on the end of this little needle right here, and you can move the needle up and down and forward and backward to bring it into focus with your glass lenses right there. And you can see in this little picture right here, you literally hold this up to sunlight or candlelight because you didn't have glass bulbs back then, and you would be able to magnify your specimen and look at it. And he was actually able to grind the lenses well enough to be able to view the microbes that are out there. And if you look at some of the stuff, it's kind of funny to look at these little primitive drawings because this is what he was actually seeing, and then trying to indicate that this little organism that he could see was actually moving around. And so he was able to actually look at these minute little animals that were in rainwater and things that he would scrape from his teeth, in addition to looking at these fibers under his microscopes. And he really was one of the first people to describe what he called animalcules, these animal molecules, these invisible organisms that were out there. And this is really the beginning of the science of microbiology, because now you can get a handle on these organisms that are out there. And this really led to what's known as the golden age of microbiology in the 1800s, because now people were starting to get more and more microscopes out there to find these different kinds of organisms. And Louis Pasteur is a famous microbiologist. Obviously, pasteurization is out there, but he's even more famous in the field of microbiology because he was one of the first people to show that bacteria were actually in the air. And so there's that idea. It's not just that you have bad air that could potentially be causing these diseases, but maybe there is this germ out there that's floating around in the air that is causing these diseases. And so he, along with a variety of other scientists, started to propose this idea of what's known as the germ theory of disease, that these invisible microorganisms that we can see with our microscopes are what's actually responsible for a lot of diseases. And a colleague of his known as Robert Koch, who actually worked in Pennsylvania, actually went a step further and showed they isolated, and his lab identified specific microbes that cause specific diseases. And the first one they did this for was our old friend anthrax. They were able to pull out the bacteria and show that it caused the disease anthrax in healthy animals that were out there. And so his lab went on to identify a whole bunch of different kinds of organisms. And this is not the lab you want to trip and fall in, because think about it. They've got all these different kinds of organisms that are causing diseases, and they want to identify what is causing it, but you definitely don't want to get that back. And that'll play a role when we're in the lab, because we really want people to start to become more familiar with this idea of we don't want contamination to be occurring, and certainly not with these organisms, but we're going to start learning some more practices in the laboratory about how you handle these different kinds of microorganisms in the lab.